At Plasmidsaurus, we offer sequencing for PCR products and other linear DNA of any size. In this video, we'll walk you through the process of preparing your sample and sending it in for sequencing. First, you'll want to choose between our linear PCR or premium PCR services. Choose our linear PCR service when you need a primer-free, any-length Amplicon alternative to traditional Sanger sequencing. Choose our premium PCR service if your sample contains mixed populations of molecules or you need higher read depth. After choosing your service, select the option corresponding to the size of the construct and the read depth that you need. Whichever service you select, the sample prep is pretty much the same. Amplify your PCR products or extract your linear double-stranded DNA using your favorite protocol. We suggest letting Plasmidsaurus handle the cleanup for you. Just click one box and we'll take care of the rest. To measure DNA concentration, the best practice is to use a qubit fluorometer. Spec methods like nanodrop might lead to overestimation of Amplicon concentration, so try to avoid those. We accept a wide range of concentrations for our linear PCR service. For premium PCR, quantify how much DNA you have, then normalize it to a concentration that depends on the length of your Amplicon. Now it's time to place your order. On the Plasmidsaurus website, choose a name for your order and a name for each sample. You'll get a 6-character order ID that you use to label your samples. For orders of 48 samples or fewer, use 8-strip tubes. The first 2 characters of your order ID go on the first 2 of each strip. The other tubes are numbered in order. For more than 48 samples, you can use 96 well plates with 200 microliter wells ordered in rows or columns. Label the front of the plate with the full 6-character order ID. Label either the first row or the first column with the sample numbers. If you're sealing your plates manually, make sure you use a plate roller and really push down hard to prevent leakage during shipping. Or even better is to use a heat sealing device if you have one. For 8-strip tubes, use a Falcon tube or some bubble wrap to protect your sample during shipping. Ship your wrapped samples inside a plastic bag. For samples in plates, wrap each plate individually and avoid stacking plates because the plates on top can damage the seals of the plates on the bottom. Print out a copy of your order form and put it inside the bag so that the QR code is visible. And you're ready to go. Drop off your samples in your nearest Plasmidsaurus drop box. Your sequences will be ready as soon as the following morning. If you have a reference sequence, you can upload it directly to the website for automatic alignment and matching. In this case, 6 out of 8 Amplicons are perfect matches, but sample number 4 here is reporting a mismatch. Scrolling down, I can check out the feature map and the base viewer showing me exactly where the mismatch occurred. Looks like I've got a single base pair mutation in the stop codon. This is going to add some junk to the end of my protein. I'll have to remake this construct before I can use it in my experiment. But I'm glad I got these sequenced and caught this problem early. That's going to save me a lot of hassle down the line. Thank you, Plasmidsaurus.